in cell differentiation and epigenetics, and now support you guys doing epigenetics here. So our webinar speaker today is Ellen. She is an expert in chromatin biology and earned her PhD from the University of Pennsylvania and completed postdoctoral studies at the Jackson Laboratory for Genomic Medicine. Here at EpiCypher, Ellen leads scientific content and technical communications for products, services, and their applications. Just as a reminder, drop your questions in the chat and we will answer them throughout, and any leftover, Ellen will be able to answer for you live. So Ellen, take it away.
All right, thanks, Jen. So my name is Ellen Winesapple, and today I'm going to be talking to you about the evolution of chromatin mapping from ChIP-seq to CUT&RUN and CUT&Tag. And just to introduce myself a bit more, I've been at EpiCypher since 2018, and I've written numerous blogs and worked with marketing on the videos to help bring our R&D and products to life. As Jen said, my background is in epigenetics research, and I'm really excited to talk to you all today about CUT&RUN and CUT&Tag. So today's webinar inspiration is a popular EpiCypher blog from a few years ago, and it's about ChIP-seq versus CUT&RUN versus CUT&Tag, and trying to figure out the best one for your experiment. So based on this, today we're going to review the problems with ChIP-seq and why it's not the best chromatin mapping assay. And then we'll talk about the advances underlying CUT&RUN and CUT&Tag and what EpiCypher has done to make these assays even more robust. And then we'll touch on key considerations for assay success and answer some of the most common questions we get from customers. And finally, we'll combine what we've learned into a case study where EpiCypher worked with the Immunological Genome Project, or ImGen, to create an epigenomic atlas of different mouse immune cell types. So let's start with why we care about chromatin mapping and epigenomics. Historically, chromatin was considered a binary open-closed system in which regions of open or accessible chromatin were associated with active gene expression, and areas of compact, closed heterochromatin were linked with gene silencing. However, with chromatin mapping technologies, scientists have uncovered a complex landscape by which chromatin structure regulates gene expression. By mapping transcription factors, histone PTMs, and other chromatin-associated proteins, ChIP-seq has helped define unique regulatory elements such as enhancers, as well as different forms of heterochromatin, all of which contribute to gene expression and cell identity. This work has also been fundamental to our understanding of both healthy tissues and disease, and holds great promise for biomarker and drug development. But with ChIP-seq, the leading chromatin mapping assay, a lot of these innovations have been limited. Much of it is due to the assay itself, which is outlined here. So in ChIP-seq, I'll just briefly go over the protocol, but in ChIP-seq, you first cross-link cells to fix the targets on chromatin, and then you lyse the cells and fragment chromatin into a bunch of smaller pieces. You use a bead-bound antibody to pull down the fragments containing your target, you purify the DNA, and then you sequence. So pretty much every step of this protocol has issues. Cross-linking introduces artifacts, which can mask your target or cause issues with sonication. Chromatin fragmentation is labor-intensive and needs to be optimized for each cell type and can be highly variable between sample preps. The IP step itself is inherently noisy, as you are using a bead-bound antibody to isolate target fragments from a pool of chromatin. Everything is sticky, and you pull down a lot of non-specific material, which requires stringent washes to remove. But at the same time, these washes remove your target fragments too. As a result, after library prep, you have to sequence very deeply to resolve signal over background. So due to these inherent problems, ChIP requires millions of cells, it's low throughput, and it generates data with variable quality. It's also prohibitively expensive, especially for large projects, due to deep sequencing. So as a result, you can't look at rare cell types or precious patient samples. You can't do large-scale experiments that require higher throughput or standardized reproducible studies that are needed for biomarkers in drug development. EpiCypher has a lot of experience with ChIP, and we did a lot of research on trying to make it more robust. In this process, we developed what we call SNAP-ChIP spike-in controls, which are panels of nucleosomes that you add to fragmented chromatin before IP. These spike-ins allow you to directly examine histone PTM antibody specificity and efficiency in your ChIP assay. So we used these nucleosome spike-ins to examine about 400 histone PTM antibodies in ChIP, focusing on common histone lysine methylation and acetylation marks. So shockingly, we found that most antibodies to PTMs perform poorly in ChIP, with more than 70% showing poor specificity, meaning that they bind other targets, or poor efficiency, meaning that they don't bind their target very well. These data are all available online at chromatinantibodies.com. But combined, these data show that ChIP-seq just isn't the right assay for the ultra-sensitive applications that the field is moving towards. So this brings us to CUT&RUN and CUT&Tag. I want to start by looking at some EpiCypher, CUT&RUN, and CUT&Tag data side by side with ENCODE ChIP-seq data. The data are really similar, but you can see right away that the run and tag data have lower background compared to ChIP-seq. Importantly, these data were generated using only 100 to 500,000 cells and just 5 to 8 million sequencing reads. This is a remarkable feat compared to ChIP-seq. When we show someone these data for the first time, they often ask, why are these strategies so much more sensitive than ChIP-seq? What is the secret? The main secret is that these assays are not ChIP. So let's dive into how these assays work, starting with CUT&RUN. All right, so CUT&RUN was first published in 2017 and was developed by Dr. Stephen Henikoff at the Fred Hutchinson Cancer Research Center. He combined immunotethering technology with a solid support to create a streamlined, sensitive chromatin mapping workflow. So in this assay, you isolate native or unfixed cells, and you couple them to concanavalin A magnetic beads. This is the solid support. The entire assay takes place in intact cells and beads, which allows for rapid processing. You then permeabilize the cells and add the antibody to your target. You then add a protein A-G micrococcal nuclease, or PAG immunase. This is the immunotethering portion of the assay. Immunase digests nucleic acids, while protein AG has immunoglobulin binding properties. So when you add PAG immunase to the reaction, it binds at the antibody-bound chromatin sites, allowing for selective cleavage at target-enriched chromatin. These cleaved fragments diffuse out of the cell, and the cells are easily removed using a magnet, effectively separating background from your target signal. You simply need to purify the DNA, prep libraries, and sequence. The whole assay from cells to libraries takes about 3 days. So here's an overview of the workflow, and right away you can see there are multiple advantages over ChIP-seq. The use of immunotethering technology and a solid support allows you to selectively capture antibody-bound chromatin, bypassing cross-linking, chromatin fragmentation, and IP. This results in an ultra-sensitive epigenomic mapping assay, where chromatin targets can be reliably profiled using 100-fold fewer cells and 10-fold reduced sequencing depths. When we were developing this assay, EpiCypher wanted to create controls to help make CUT&RUN as robust as possible. So we adapted our nucleosome spike-in technology from ChIP for CUT&RUN, resulting in SNAP-Katana spike-in controls. So these nucleosome panels are bound to magnetic beads and are added to cells just before the antibody is added. Your antibody will capture the on-target spike-in alongside target chromatin, and the spike-ins can be detected in your sequencing data, allowing for analysis of histone PTM antibody specificity and assay success. So with this addition, you have all the benefits of the standard CUT&RUN workflow, plus defined quantitative controls. All right, and this is its sister technology, CUT&Tag, which has many of the same benefits, but works slightly differently than CUT&RUN. Instead of using a PAG immunase, CUT&Tag uses a protein AG fused to TN5. So the TN5 will both cleave antibody-bound chromatin and add sequencing adapters, bypassing library prep for a streamlined 2-day workflow. And EpiCypher has adapted the protocol to go from cells to DNA in one tube, further reducing hands-on time. We have adapted the SNAP-Katana spike-in controls for CUT&Tag workflows as well, providing an additional quantitative control. So again, CUT&Tag generates highly reproducible profiles from low cell inputs and sequencing depths, similar to CUT&RUN, but it's also been used to profile single cells in the literature, making CUT&Tag a really exciting technology. So when Epicipher was developing these assays, we realized immediately that the improved sensitivity, throughput, and lower costs could be highly empowering, allowing epigenomics to go places that were impossible using ChIP-seq. CUT&RUN and CUT&Tag could finally allow scientists to examine these rare cell types, do scaled clinical profiling, and harness epigenomics for biomarker and drug development in a reliable manner. So we kept this philosophy of being able to do more in mind when we developed our CUT&RUN and CUT&Tag kits. Each kit is designed for 48 reactions, which is among the larger kit sizes on the market, but also has the lowest price per reaction. We streamlined our kit protocols by making them compatible with 8-strip tubes and magnets, and each kit includes robust controls to help ensure experimental success. We also pride ourselves on our excellent technical support, and we're building an online tech support center for on-demand guidance. So now that we've reviewed the basics, let's go into some key considerations for assay success. First off, we'll touch on the topic of our inspiration blog: which assay is right for me, CUT&RUN or CUT&Tag? So the real answer is that it depends on your experiment. There are three main things we look at. First of all is, what is your target? The second is, how many cells do you have? And the third is, what is your experience with genomics assays? And either way, we have all the assays, kits, and controls that you need to be successful. But first off, let's look at targets. So our Katana CUT&RUN assays work for pretty much every target we've tested, including proteins that have been inaccessible by ChIP-seq, such as chromatin remodeling enzymes. Here we're showing SMARCA4 or BRG1. But yeah, we're looking at a bunch of different targets here: transcription factors, chromatin readers and writers, histone modifications. CUT&RUN is robust for everything. On the other hand, Katana CUT&Tag is generally only recommended for histone modifications. And here is CUT&Tag data from K562 cells looking at a different array of histone modifications. We do want to note that we have tested CUT&Tag for transcription factors. We tested 18 different transcription factor antibodies that we had validated for CUT&RUN, and only 3 of them were successful in CUT&Tag. So it really is most robust for histone modifications. All right, and the second question is, how many cells do you have? So for CUT&RUN, we recommend using 500,000 native or unfixed cells per reaction, but you can get robust signal down to 5,000 cells for select targets. And for CUT&Tag, we recommend using 100,000 native nuclei per reaction. We do recommend using nuclei. It is robust down to 10,000 nuclei for some targets. I also want to note that using more nuclei isn't recommended for CUT&Tag. We don't recommend using more than 100,000 because then that just causes some issues with the assay itself. And we also get often asked, what if I can't meet the ideal cell numbers? Lower cell numbers can work, but there are some considerations. So for low abundance targets, such as some transcription factors, it can be challenging to generate enough yields from low cell numbers. It's definitely something you want to keep in mind. For low cell numbers or low abundance targets, you also want to make sure you are using a highly efficient antibody, meaning it reliably recognizes and binds its target epitope. Antibodies that work well at standard cell inputs may fail at lower cell numbers due to poor efficiency. And this is best illustrated by looking at an example. Here we are doing CUT&RUN for H3K27ME3 using a high and low efficiency antibody. So both show good signal at the recommended cell depth of 500,000 cells. The high efficiency antibody is robust down to 10,000 cells, while the low efficiency antibodies really falls apart as the cell input decreases. We have seen this happen for multiple antibodies we've through our screenings, and it's just a fact of that a lot of antibodies aren't perfect. As a side note, Epicipher, our histone PTM antibodies, are always validated for high efficiency by testing at our standard cell numbers and then tenfold below that, ensuring reliable antibody performance across a range of cell inputs. All right, and the last question looking at cut and run or cut and tag is, what is your experience with genomics assays? So for most users, we recommend cut and run, but for highly experienced users, or if you're doing something very specific that requires TN5, cut and tag might be recommended. So when we're thinking about epigenomics and skill, it's almost easier to compare it to something we do every day. So I like to think about driving. Most of us drive almost every day. We've been doing it since we were teenagers. For the most part, we're safe and we know how to do it. A Formula One driver also drives, and at baseline, we are all drivers, but they have years of experience driving unique cars under high speed and intensity that enable them to drive their race cars. You and I, we could drive their cars, but we'd have a hard time maybe keeping on the road and run into potential issues. So this is similar to the cut and run versus cut and tag debate. We can all use cut and tag, but without that extra level of expertise, you have an increased likelihood of issues and just in general having a hard time getting the data that you need. So if you're still unsure, reach out to us at techsupport@epicipher.com and we can help find the right solution for you. All right, so this is a summary of everything we've talked about in this section. Definitely take a screenshot. I would say this is the main takeaway from this section of the talk. So again, choosing between cut and run and cut and tag, you definitely want to use cut and run if you're looking at chromatin-associated proteins such as transcription factors. Cut and run, the ideal input is 500,000, while for cut and tag, 100,000 is the ideal input. Cut and tag can go lower. In the literature, there have been numerous single cell cut and tag papers. So if you're interested in going super low input with cut and tag, definitely check out those papers. Processing time, both cut and run and cut and tag are very similar. Cut and tag is slightly faster because you're bypassing standard library prep. In both cases, if you want to get started, Epicipher has kits for each assay, as well as enzymes, antibodies, and all the controls you could need. Visit epicipher.com or email techsupport@epicipher.com for help. All right, and the next consideration for assay success is how do you optimize these assays for your project? And this really relates to, will the assay work for my sample type? And how should you select the best antibody? So is my sample type compatible with cut and run and cut and tag? Chances are it is. We have protocol adaptations for almost everything. We have adaptations for both fresh and frozen cells and nuclei, as well as lightly cross-linked samples. I do want to mention that for the cross-linking, we don't recommend it in most cases because it usually doesn't help. If you're going to test cross-linking, definitely follow our protocol for light cross-linking, not heavy cross-linking as in ChIP, and always test side by side with unfixed samples just to see what that comparison is. We have adaptations for adherent cell lines as well, and we've done both cut and run and cut and tag with primary cells, and we have used drug-treated samples. We can get robust data from pretty much any of these sample types. We've also begun looking at optimizing sample prep for tissue for cut and run. So here we used frozen biopsies from the intestine of patients with and without Crohn's disease. And we used an intestinal cell line as a control, the CACO2 intestinal cell line. And we profiled a variety of targets, H3K4ME3, which marks active promoters, the repressive mark H3K27ME3, and a transcription factor CTCF. IgG is included just as background to measure background. And we see that we get high quality profiles from the frozen tissues that look very similar to the fresh cells, which is exactly what we would want to see. There are some key lessons that we can take away from this experience. First of all, processing to a single cell suspension is critical for cut and run and cut and tag from tissues. You want to avoid cell clumping, which can complicate the downstream assay. In many cases, we actually recommend nuclei extraction, which can help avoid this stickiness inherent to some cell types. We are continuing to work on tissue prep methods for cut and run and cut and tag, and we hope to have more standardized methods available soon. So definitely watch our website and social media for more information. Okay, so now that you've optimized it for your sample type, how can you make sure you select the best antibody? EpiCypher has screened more than 1,000 antibodies in cut and run, and we can definitely help you find the antibody you need. Here I'll go through how EpiCypher tests and figures out which antibodies we want to use for our experiments. So first of all, this is one of the most common questions we get. ChIP antibodies are not guaranteed to work in cut and run and cut and tag. Again, cut and run and cut and tag are very distinct assays from ChIP. So you can test ChIP antibodies and see if they'll work, but we always recommend testing side by side with additional antibodies. So picking the best antibody, we recommend, again, sourcing 3 to 5 different antibodies, ideally from different vendors targeting different epitopes. EpiCypher offers a lot of antibodies that are already validated for cut and run. Definitely check out our list. And again, if we don't have something that fits your experimental needs, reach out, because we've screened like 1,000 antibodies and we can definitely help guide you in the right direction. So once you've sourced your antibodies, you want to test them side by side in cut and run or cut and tag. If you're looking at antibodies targeting histone lysine methylation marks, include SNAP Katana spike-ins in your reactions. This will help tell you how specific your antibody is in the assay. All right, and once you've performed your experiments, you want to analyze the data side by side and select the best antibody based on yields, signal to noise, and expected target enrichment patterns. So let's look at an example from EpiCypher's own work. Here we sourced 5 different P53 antibodies, and this is just an example of the data. And this is one of a great example of why it's important to source multiple antibodies to your targets, because of the 5 we sourced, only 1 generated really high quality data. You can see that antibody 1, you can see clear peaks, whereas with the other 4 antibodies, it's pretty much nothing, just a little bit of background. So this is just an example of how we test antibodies at EpiCypher. And for our histone PTM antibodies, we have found that identifying really specific antibodies has enabled new insights into PTM functions. So as we said before, a lot of histone PTM antibodies are cross-reactive. They'll most commonly cross-react with PTMs that are very similar or at similar locations. So in the past, if you're looking at something like H3K27 methylation, a lot of these antibodies would react to all 3 lysine marks. Now that we have really high quality antibodies that are highly specific for the ME1, ME2, and ME3 states, we're seeing these differences that weren't possible using poorly validated antibodies. And we're looking into the biological function of these differential enrichment patterns between H3K27ME1 and H3K27ME3. And we're really excited about where this research might go. All right. And of course, the number one question, how do I know if my experiment worked? All right, so controls are critical to Katana, cut and run, and cut and tag success. Each of our kits and manuals include multiple quality control checks to support your workflow. The first quality control check is always to confirm high quality sample prep. This is critical for assay success. If you put bad cells in. If your cells have a ton of lysis, you're not going to get very high quality data. We always check the number of cells, making sure that we're starting with that standard amount if possible. You want highly viable cells, ideally more than 80%. You want to check and make sure that your cells have the expected morphology. If you're isolating nuclei, you want to check those under the microscope, make sure that your nuclei were fully isolated and there aren't dead cells hanging out or a lot of debris. And then we also always check binding to beads because if your sample isn't bound to the beads, you could lose a lot in the washes. A note on cell viability. I will say that for certain samples, it's difficult to have high viability. Some primary cells are very difficult to culture and you don't get more than maybe 70% viability. It's important to focus on the optimal viability for your cell type and just have your samples be as high quality as possible. All right, and we always include positive and negative control reactions. These are included in each of our kits as well. So we include the positive and negative control reactions. These reactions are always spiked with our SNAP Katana K-Metstat panel of spike-in controls. And so the reason that we love these controls is that they can validate the workflow and boost confidence in data, but they can also flag bad workflows. So basically, if your control reactions aren't working well, then you know that there could be potential issues with the workflow. So in this example, the top panel is data from your spike-in controls, and the bottom panel is sequencing tracks. And we had 3 different B cell samples, and we profiled H3K4ME3 with our validated antibody and added the K-Metstat panel of spike-in controls. So samples 1 and 2, the spike-in data looked fantastic. So you're looking at the percent recovery of each nucleosome in the panel relative to the on target. So you should only see basically one blue square, which is the H3K4ME3 spike-in. Now, sample 3, you're seeing a lot of recovery of off-target spike-ins and just a generally poor signal to noise when you normalize even to your on target. So this is suggesting that something is off. And then when you look at the actual sequencing tracks, you can see that samples 1 and 2 generated beautiful data, but sample 3, the tracks, the signal is very poor and there's basically no data here. So combined, these data suggest that our workflow is fine because samples 1 and 2 are successful, but there is potentially something wrong with our sample prep. And indeed, we found out that we had started with far fewer cells than what we thought we had. So our kit manuals describe these controls in detail, how to use the results to troubleshoot, how to use the results to confirm your workflows, and how to analyze the data. So definitely don't skip that portion of the experiment. And finally, you always want to check the fragment distribution of your purified sequencing libraries. In general, and this is all described in our kit manuals, you'll want to see library enrichment at around 300 base pairs. This is the mononucleosome fragments. And this is the best indicator of success before sequencing. Importantly for cut and run, make sure that you're only looking on the tape station or bioanalyzer after library prep. Usually the yields from cut and run are too low to look before library prep. So only perform tape station or bioanalyzer for your final purified sequencing libraries. All right, so now that we've sort of covered how to optimize and design what makes a good cut and run and cut and tag experiment, let's apply this to our case study where Epicipher worked with Imgen to develop the epigenomic atlas of different mouse immune cell types. So to give a little bit of background, Imgen is a multi-site consortium whose goal is to generate a full molecular atlas of the mouse immune system, from gene expression and chromatin profiles to proteomic analysis. This data will be extremely helpful in understanding the inner workings of the immune system and identify new strategies for autoimmune and inflammatory disorders and cancer. Imgen approached EpiCypher for chromatin profiling analysis because ChIP-seq really lacked the sensitivity needed for many of the cell types they wanted to analyze. So here is what they were requesting. They wanted to analyze 80 to 100 different mouse immune cell types, many of them isolated by FACS. And they wanted to look at 8 distinct chromatin targets representing various chromatin elements. So H3K27 acetylation would be marking enhancers, H3K4ME3 would be marking active promoters. You have a marker of heterochromatin, H3K36ME3 marks active gene bodies, and then they also wanted to profile CTCF. So right away, we knew that CUT&RUN would be the preferred assay for this project due to its robustness across both PTM and transcription factor targets. But this is a challenging project with more than 80 cell types to optimize for and at the lower sensitivity limit of the assay, not to mention the sheer throughput needed to process this number of samples. There were a lot of factors to consider. Ultimately, we reasoned that automation was the only way that we could make a project of this size robust and reliable, but it would take multiple controls and careful optimization to be successful. So let's dive into this a little bit. In particular, to analyze low cell numbers, we needed antibodies that are highly specific and have high efficiency for their target, as we discussed on the previous section. So we used the SNAP-Katana nucleosome spike-in controls to identify highly specific antibodies for each of the 8 targets. So shown here is a heat map of antibodies we had previously identified to lysine methylation targets using our K-METSTAT spike-in panel. And data for each antibody are shown by row, and recovery of nucleosome is normalized to the on-target PTM. So you can see that these are each highly specific. We then took our highly specific antibodies and we tested them in cell titrations using K562 cells. For each target, we found antibodies that were robust down to 10,000 cells, which is a 50x reduction from the standard 500,000 cell input. And then we also optimized the entire workflow for automation in 96 well plates. So notably for H3K36ME3, we had to source 10 different antibodies to find one that met our stringent criteria, which highlights the weaknesses of many PTM antibodies and the importance of testing against defined controls. So these high quality antibodies were essential once we moved to profiling primary immune cells. This is data for primary B cells, 10,000 per reaction with 2 replicates for each target. So you can see that the results are highly consistent, even at these strikingly low cell inputs, highlighting the reproducibility of this platform. And of the 19 plates we ran for the Imgen project, only 1 failed our quality control metrics, demonstrating the robustness of our workflow. So let's go over some of the controls and optimizations along the way. So one of the factors that we had to think about for the Imgen project was that we were processing many different cell types and they're coming from many different labs across the country. So the first thing we wanted to do was optimize our sample prep. So we tested native versus cross-linking for different cell types and for each of the targets. And then we also ended up optimizing a new nuclei extraction buffer and protocol. This allowed us, we ended up going with nuclei versus cell prep because it allowed us to have more standardized processes and get quality nuclei shipped from all these different consortia sites. And so whenever we received samples, the first thing we always did was confirm the nuclei count and look at their morphology, make sure there wasn't a ton of lysed cell debris in the prep. And then after we bound the nuclei to beads, we would always confirm bead binding. We then had controls for each cut-and-run reaction. So we added SNAP-Katana spike-in controls to monitor antibody specificity and to make sure the reactions were working as expected. And then we also added E. coli spike-in DNA. That's added before library prep. So it's basically to monitor the success and efficiency of library prep. And because we were running multiple plates, we ran a total of 19 different plates, we needed a way to compare the success across plates. And we are also analyzing about 100 different cell types. So we needed something standard that would allow us to look at each plate side by side. So what we ended up doing was using a control mouse cell line, NIH3T3 cells, and we ran them in each plate with all 8 antibodies. These reactions also included our SNAP-Katana spike-ins and E. coli DNA. And this allowed us, our control reactions were one of the keys to our experimental success. 99% of our control reactions passed our QC metrics. And that, again, was crucial to the success of this project. So this is a preliminary view of some auto cut and run data from the ImGen project. We're looking at 3 different innate immune cell types, granulocytes, type 3 innate lymphoid cells, and natural killer cells. And we can already see that there are some differences in peak patterns among these fairly related cell types. And we're excited to start digging into the data a little bit deeper because this type of information is so important to understanding the different functions of these cells. All right, so to summarize, the entire ImGen project is really a case study in the types of projects CUT&RUN and CUT&Tag are allowing people to do. The ImGen project, simply put, would not have been possible with ChIP-seq. But even if you're not doing a massive experiment with hundreds of sample types and reactions, there are ways that CUT&RUN and CUT&Tag can benefit your research. So we challenge you to think about what you could do with this technology. Epicipher is continuing to work in new areas using these immunotethering tools. We're developing new multi-omic assays looking at histone modifications and transcription factors alongside DNA methylation. And we're also working on chromatin accessibility assays for FFPE samples using the CUT&Tag related assay ChIP-seq. We do want to note that ChIP-seq assays are a feature of our CUT&Tag kits, and everything that you would need for the ChIP-seq assay comes with the CUT&Tag kit. Okay, so we've reached the end of the talk. So our general take-home messages, CUT&RUN is our user-friendly go-to assay that works for most targets and samples. And CUT&Tag is really designed for low-input histone PTN mapping, and it's for expert-level users. In both cases, we have the kits that you need to get started. And we also have numerous antibodies and accessory reagents available at epicipher.com. If you have questions about your experiment, how to get started, definitely give us an email, techsupport@epicipher.com. And with that, I just want to acknowledge everyone at Epicipher. I can't think of a single person who didn't contribute to this talk in some way. And I also want to thank ImGen for allowing us to present on this project. Yeah, with that, I'll take any questions.
Great, thank you, Ellen, for a fantastic webinar. So there's been plenty of questions in the chat, which we've been answering, and feel free to add additional ones. Some that we didn't get to is, so what would be the ideal positive and negative control for transcription factor experiments in CUT&RUN?
Oh, that's a great question. Well, you definitely want to include IgG as a control just to assess background. I would say for transcription factors, the ideal control, you'd probably want to make sure you're testing multiple antibodies to make sure that you're getting the most robust enrichment. And then I would think if you have access to a knockout cell line to make sure your antibody is really specific or is really robust, that could also be useful. I don't think I have any other ideal controls for a transcription factor other than maybe related proteins and histone modifications that could be targeting the same type of pathway.
Yeah, that's great. And I know from the tech support angle, sometimes we will recommend including CT-CF as well, because that can be extremely robust. And we have an antibody to it that works very well under many conditions. Yeah.
Yeah, that's a really good point.
So let's see. Got quite a few going on here. It looks like we've got a couple people typing. So one that we have is, does CUT&RUN and CUT&Tag work well for dense heterochromatic regions?
Oh yeah, absolutely. I actually have a slide about that. I'm going to go back to full screen so you can see it. So it definitely, both CUT&RUN and CUT&Tag work well for repressive heterochromatin. This is data from a paper with our collaborator, Ari Melnick, from a few years ago in Nature. And so this is looking at CUT&RUN in mouse germinal center B cells. And we're getting robust signal for H3K9 methylation, even across areas that are generally very difficult to map, and DAC exclusion list regions. And then we're also getting strong signal for H3K27 and E3 as well.
Great, thank you. So one from the chat, it's from Tegan. I'd love to hear what kind of analysis parameters we use in standard experiments and whether that has to be modified for different cell types.
Do you mean like data analysis?
I believe, yes, that would be data analysis, but could you also comment on, does the actual experiment workflow have to be modified with different cell types?
With different cell types, yeah. So you may have to modify your sample prep for different cell types. So we have a lot of modifications depending on if you're working with adherent cells. We have methods that you can basically dislodge them from the cell culture plate without disrupting the cellular integrity. We have modifications if oftentimes if you're using primary cells or immune cells or tissues, we do suggest using nuclei, and we have extensive nuclei prep protocols. There are some other considerations, and I would say if you're really concerned, I would look at what the protocol modifications we have available for sample prep, and then if there's nothing that matches your needs, I would reach out to tech support. As far as the data analysis goes, we will sometimes modify the stringency of like our peak widths depending on the target. That's pretty much standard across a lot of chromatin mapping workflows. I would say in general, we follow the protocol for data analysis that's in our manual, and it's on the tech support center. And it's quite similar to ChIP in a lot of ways.
Great, thank you. And they did clarify it was on peak calling, so I think.
Oh yeah. So yeah, we use for targets like H3K27 ME3, we'll use like I believe we use Sicer, and we set a broader peak window. And then for targets like H3K4 ME3 that have these super sharp peaks, we'll use a narrower window to define those peaks.
Okay, so we just got one that is cell prep versus nuclei for the assay. How much of a difference does that make in sample quality and data quality?
It really depends on your starting sample. I hate to say it depends because that's not a very satisfying answer, but yeah, it really does depend because sometimes if you're using a cell sample, like let's say you're using an adherent cell line, and even after harvesting your cells, they're just very sticky, and they're not going to a single cell suspension, which is really important for CUT&RUN and CUT&Tag. Using nuclei in that case would be preferred because then you're getting rid of the cell membrane that's causing all those issues. I would say there are some caveats to using nuclei. Some people lose a lot of cells during the nuclei extraction. So if you're worried about that, I would say use cells. So that's kind of like the pluses and minuses of it. If there's anything else you think to add to that, Jen.
I think you covered it fairly well. Ultimately, it is the prep, the quality of the sample is the most important, and then the data is going to be pretty comparable. So a question about the ImGen project specifically, how were the cells shipped to us? How were they stored? Did we process them immediately upon receipt?
All right, I was not personally involved with doing any of the bench work for the ImGen project, but to my understanding, they were shipped to us on probably dry ice, and they were shipped to us, the labs prepped the nuclei using our standardized methods. They shipped them to us on dry ice. Once we received them, we analyzed them, we basically counted the nuclei, made sure that they were intact, that they hadn't lysed or anything during shipment. And then I think we probably stored them until we were ready to do the cut and run experiment.
Mm-hmm. Yep. And that's typically how we do services. We now actually offer a cut and run service for kind of anyone who would want it, and we'll receive the samples, store them until we're ready to process, if we receive multiple or not. Let's see. We have some more questions. Oh, looks like that one got cut off. So another question, can we use cut and run and cut and tag for drug treated samples?
Oh yeah, absolutely. I actually have an extra slide about this as well, because I wanted to include all my data and I couldn't include all of it. So this is an example of a cut and run experiment where we use drugs treated with Tazverik, which is just tazemetostat. It's a clinically approved EZH2 inhibitor. So just to remind folks about how EZH2 works, EZH2 is a histone lysine methyltransferase. It catalyzes H3K27Me3. This is a repressive mark. So basically, EZH2 will bind the H3 tail, generate this mark, and that leads to gene repression. So when you use this drug, it blocks EZH2 from generating this mark, and you should lose H3K27 methylation over time, and theoretically, your genes will turn back on as well. So we treated cells with either vehicle or DMSO, 1 micromolar of drug and 5 micromolar of the inhibitor. And then we looked at cut and run for the drug target, EZH2, as well as the enzyme product, which is H3K27Me3. And then we also looked at active promoters marked by H3K4Me3. And so you definitely see decreased localization of the drug target compared to baseline, and also less H3K27 methylation. Interestingly, at some locations, including the one shown here, we did see like reactivation of genes as denoted by H3K4 methylation. This wasn't at every gene we analyzed, but it was an interesting observation.
I muted myself. Sorry about that. So what are the challenges in using transcription factors in cut and tag? Why can't we do that?
That's a great question. Cut and tag, in general, I think a lot of transcription factors are low abundance targets. And so there's just not a ton of transcription factor bound in your genome. As a result, the tagmentation reaction sometimes is not efficient enough for these low abundance targets to get high enough yields for sequencing. That's our best guess right now. We are working on making these protocols more robust, not just for transcription factors, but for all kinds of targets. In general, I think the tagmentation is it's an amazing enzymatic reaction, but it's because of having to insert the adapters at both ends and making sure that you have the two different sequencing adapters. The tagmentation efficiency can sometimes be it's just hard to get the right combination of adapters, basically, for PCR amplification.
Another question related to cut and tag: is it possible to use drug-treated samples in cut and tag?
Yeah, I would think so. Yeah, I think Yeah, the data that I have here is for cut and run, but I'm pretty sure that you could use cut and tag. You could use drug treated samples in cut and tag as well.
And anecdotally, I've definitely spoken to researchers who are doing that. So it is feasible.
Yeah.
Absolutely. Yeah. Just would have to be studying something that would target PTMs instead of a transcription factor. So another question related to transcription factors, is it possible to make a CTCF spike-in control?
Yes. Theoretically, yes. If you wanted it to be like our nucleosome spike-in controls, we would basically have to design whatever epitope the CTCF antibody is bound to, and put that, like, attach that to the nucleosome. That's actually what we did to validate our CTCF antibody for the ImGen project, because we wanted all of them to be validated by the nucleosome spike-in controls. Doing that for every transcription factor antibody would be almost impossible, but that is how you would do it.
Thank you. Another spike-in control question: can we add E. coli or any other sort of DNA as a carrier for low input cut and run or cut and tag?
You can. It's not something we typically do at Epicipher, because we prefer to use our SnapKatana spike-in controls. We do use E. coli for our cut and run experiments. We have added it to basically towards the end of the protocol after immunase activation, and we use it as a control for library prep, and you can use it for sequencing normalization. For cut and tag, we haven't optimized the E. coli spike-in quite yet, so we couldn't provide guidance on that. But there are examples in the literature where people have used like E. coli. I will say, like, our preparations of our enzymes are incredibly pure, and there's really no E. coli like carryover in them. So if you wanted to do something like that, you would have to add E. coli DNA. It's not present in our purified enzymes.
Great, thank you. Let's see. Looks like we've got Nathan answering a question for us. Let's see. So what are the typical reasons an antibody would fail in an experiment?
There are a few. It depends on if you're looking at a PTM target or a transcription factor. If you're looking at a histone PTM, if we're able to use our SnapKatana spike-in controls, we have strict metrics on cross-reactivity. So if it's binding off-target PTMs more than 20% relative to the stated target, that would be a reason for that antibody to fail. If it's not generating sufficient signal over background, basically if it looks exactly like your IgG control, then that would be a reason your antibody would fail. For a transcription factor, you would want to make sure that if there is a standard motif for that transcription factor, you would want to make sure that you're enriching for it. If you're not enriching at these expected motifs, that's potentially an issue, or it could be an issue with your cells being perhaps stimulated properly. But this is why we definitely always want to test multiple antibodies to these targets to find the one that's most robust.
Sounds good. And I've definitely seen instances where cell prep can play a role into that as well, coming from the tech support angle. Let's see. Looks like we've got our team answering. Yes, this is something we see a lot in tech support. Cell or nuclei clumping is one of the major problems. Do you have any suggestions to reduce the clumping?
Yeah. So I know that there are some people that will add a low percent BSA to their buffers. That can help reduce clumping. I will say, in general, starting with really high quality samples is the most important thing to helping reduce cell clumping. A lot of times cell clumping happens because there's a lot of cell lysis, or if you're just not pipetting your samples very carefully. Is there anything from your experience you want to add there, Jen?
Yeah, I've definitely spoken with researchers working in different immune cells, different primary tissues that seem to be a bit more fragile. So the cells tend to break up, and even during that nuclei extraction, so we have a protocol for that, which has a 10-minute incubation, and that does seem to be a bit too intense. The nuclei can continue to break up and gum up and create clumping. So you can actually use a decreased amount of time during that incubation, which does seem to help. Typically, it is, as you said, a cell problem. So either trying a different isolation method, or if they are adherent cells, maybe decrease the time exposed to trypsin. There's lots of different tips and tricks, and we can definitely follow up with you to help decrease that clumping.
Yeah, and I will say another thing about sample quality is in our manuals, we really stress that you need to check your sample quality at at least three different points. So you check your cells, you count them and look at their viability and integrity at the start of the experiment, like right when you're harvesting them. And then you do several washes in a cut and run or cut and tag. In cut and run, you do several washes in a cut and run buffer before you even bind them to the connate beads. We always recommend looking at your cells again before adding them to the beads to make sure that they're okay. A lot of cells don't like the cut and run buffers for whatever reason. So if all of a sudden, like all of your cells are lysing, or the viability as just the morphology or viability is just like changed dramatically, you may want to consider like reaching out to us, and we can help troubleshoot that. Because I feel like that's a big problem. Like sometimes people just check at the start of their workflow and not before and after bead binding.
Yeah, that is correct. We definitely see that. And it's a really helpful metric to take a look at. Another question for you. Someone who is using both cut and run and cut and tag and is seeing adapter, primer, and dimer issues during both with lower input, so 10,000 to 5,000 cells. Do you have any suggestions?
Yeah, so that is unfortunately really common when you're using lower cell inputs, because especially with the adapter dimers, you simply have less yield for the amount of adapter in there. You can, we actually have a new protocol using SPRY technology to do size selection for those libraries. It's in our quick cleanup DNA purification kit manual. If you follow up, we can send you that protocol to help size select. I will say it depends on, for size selection, if it's, do we say like if it's more than like 5% of your library, you should do the size selection. So it depends on how bad the contamination is. As far as strategies to reduce it, for cut and run, making sure that during the adapter ligation steps, you want to keep everything as cold as possible. Making sure you follow all the instructions in the kits really helps. For cut and tag, there really isn't a way to eliminate primer dimers, especially when you're using such low inputs. So the best strategy would be to either try to use more cells, or just plan on having to pull your libraries and then do adapter cleanup or primer dimer cleanup.
Great, thank you. And yeah, we'll follow up. We've noted your names if there's additional information we can provide, resources, and provide some tech support. Some are definitely more thorough questions than we can answer in the webinar, but that does bring us to time. So thank you all for joining us. We hope you enjoyed, and there will be a recording sent out shortly after the webinar for you guys to watch.
Yeah, thank you so much, everyone.